we cannot really make it. Hindi naman natin pwedeng i-shorten yan at sabihin ko na itong fixatives lang na to ang dapat nyong i-memorize. Hindi pwede. So, dapat ay, natapos ko ba, dun sa discussion na binigay ko, natapos ko yung buong fixatives? Please enlighten me. Saan ako saan ako nagsimula dun sa binigay ko na video? Baka may hindi ako na-discuss dun na part.
According to composition, ma'am.
According to composition.
Ma'am, yung last part po is glutaraldehyde.
OMG. So, hindi pa kompleto yung binigay ko sa inyo. So, ang na-discuss doon sa video is
Types of
according
fixatives yung start po, ma'am, until
types of
glutaraldehyde.
fixatives. According to composition.
Yes po.
So, you know na the different fixatives, like for example, your simple, your compound, na-discuss doon, ano?
Yes po.
And then, yung according to action, we have your microanatomical, cytological, nuclear, histochemical.
Yes po.
Okay. And then, the difference between cytological and nuclear. Okay. So, hanggang glutaraldehyde lang pala ang natapos doon.
Yes po, ma'am.
So, ibig sabihin, may metallic fixatives pa. Oh, I see. Okay, sige. Sige, I will upload the metallic fixatives later na lang. Okay? So, that's why, kaya nga dapat tapusin nyo na, natapos nyo na, or patapos na dapat yung activities nyo. So that tomorrow, in case na hindi nyo pa siya tapos, I mean, in case na hindi nyo mapanood yung videos ko before our meeting tomorrow, so I can probably give the rest of the time for you to review for your lecture quiz on Saturday. Okay? So, I will also be uploading yung decalcification video. Okay, yung decalcification video na natin tomorrow. Okay, hindi ko pa siya in-upload kasi nandun yung sagot sa QFR ninyo. Okay, some of the answers are in there. So that's why, of course, I will not upload it. Okay, kasi baka mamaya ang reference na gamitin nyo doon, video of Ma'am Gili. Okay? Dapat di kayo naglalagay ng ganun na reference kahit nasa module, hindi nyo dapat siya nilalagay na reference. Okay, so anyway, enough of that. Yes, and let's proceed to our discussion for today. Okay, so again, of course, since this is the introduction part, so let us try to define what is fixation. Okay, so in the preserved tissue preparation and examination, so the first step that we have to perform in order for us to start the tissue processing is, of course, your fixation. Okay, so you should know by heart the different steps which are needed in order for us to process your preserved tissue. Okay, so in this case, take note that your fixation is considered to be the first, okay, and it says here the most critical step in tissue processing. So of course, self-explanatory naman why it is considered to be the first, because it's basically the first. Okay, but why is it considered to be the most critical step in tissue processing? This is because, remember that when we examine, or when the pathologist examines your prepared tissue section later on, of course, the pathologist would like to examine the different cells and structures in as lifelike manner as possible. Okay, so that means, the architectural pattern, okay, or the morphology of your tissues. So that would include, of course, your cells as well as the different components of your extracellular matrix. They have to be preserved, okay, habang buhay pa sila. Kaya nga ang nakalagay doon ay in as lifelike manner as possible. We don't want, okay, we don't want our cells, we don't want our extracellular matrix to undergo disintegration. Why? Because you will not be able to show to the pathologist later on what are the different structures which are necessary for the diagnosis of your patient. Okay? So say for example, yung cell, 'di ba? Yung cell, it's supposed to be alive. It's supposed to be normal. Would you still be able, or would the pathologist be able to recognize it as normal if you were not able to carry out fixation properly? Okay, so hindi na, kasi nag-disintegrate na yung cell eh. Tama? So dapat yun nga, in as lifelike manner as possible. So that's why it is the most critical step. So the diagnostic quality of your tissue sections later on, it is only as good as how you were able to carry the fixation process. So if, for example, you were not able to carry the fixation process properly, then it would have a significant effect on the appearance, okay, of the different cells, of the different structures in the extracellular matrix of your tissue sections. So that's why siya ang tinaguriang most critical step in tissue processing, okay? So take note that the process of fixation, it has to be performed immediately. Why? Because as soon as you remove cells or you remove tissues from the body, it will automatically undergo disintegration. Okay, so it will automatically undergo disintegration. Remember, okay, remember ha, what is needed by your cells and your tissues in order for them to survive? Ano ang kailangan nila for them to survive? Di ba? Ang ingay ng mga bata, ng mga aso. Okay. I hope you don't mind Itong mga batang to, sigawan ko na yata. Ano? Ano ang kailangan? Okay, oxygen, which is carried by what? What carries oxygen to the different cells and tissues of your body?
RBC.
Okay, RBC. And your RBC, it's part of the circulation. Okay, so that means vascular perfusion. Diba? Tama ba ako? Are you familiar with the term vascular perfusion, or the vascularization of your tissues and organs would serve as a means for your tissues, for your cells in particular, to get nutrients from the circulation. Tama? Okay. So the nutrients which are primarily needed by your cells, for example, we have your oxygen, we have your glucose. So all of these are provided by the circulation. Now, if you remove your tissue or your organ from the body, what are you stopping? Ano ang pinagkakait mo, kumbaga, doon sa ating tissue or sa ating cells? It's the circulation. Tama. Nawawalan siya ng perfusion, nawawalan siya ng source. Okay? Nawawalan siya ng source ng nutrients. Kasi 'di ba, wala nang dugong dumadaloy doon sa ating ano pangalan nito, doon sa ating cells or tissues. So you are taking away its supply, okay, of oxygen and other necessary nutrients for it to survive. So without these nutrients, without this constant supply provided by the circulation, of course, your cells and the different components of your extracellular matrix, so it will undergo disintegration. Okay, so we cannot really do anything about it. So that's why you have to perform fixation immediately. So as soon as you remove, or the surgeon perhaps removes the organ from the body, so it has to be immersed in the fixative. Okay, agad-agad yan. Alright? So if, for example, it's in the case of your autopsy materials naman, ganun pa din. Okay, so kailan mo ba nilalagyan ng formalin yung isang deceased body? Okay, so inilalagyan natin siya ng formalin as soon as Okay? As soon as death is already confirmed. Okay? Syempre, siguraduhin mo munang patay yung tao. Bago ka mag-fix ng isang katawan ng tao. Okay? Yes. So you have to establish death first. And this will be taken to a special place in the hospital, perhaps in the morgue of the hospital, and it is in this place that fixation is carried out. So as soon as death is already confirmed, you have to fix, okay, the entire body. So again, pag surgical materials naman, as soon as the material is removed from the body, it should be fixed. Okay, so that's how immediate it is. Okay, so ganon ang ibig sabihin ng immediately. Okay, so there are two purposes of fixation. So the primary purpose of your fixation is for preservation. Okay, so when we talk about the preservation of your tissues, there are two things that you have to preserve. So number one is the morphology. Okay, morphology of your cells. Okay, morphology of the fibers perhaps that are in the extracellular matrices. Okay, and another would be the architectural the architectural pattern of your tissue constituents. How are they arranged? Okay, so hindi naman pwedeng cells lang ang ma-preserve. Hindi din pwedeng fibers lang ang ma-preserve. Okay, may cases po kasi na preserve ang ating tissue constituents, pero the architectural pattern is not preserved naman. Okay, so hindi din maganda pag ganon, hindi din pwede na ganon. So, ibig sabihin ang ibig sabihin kasi ng architectural pattern, it is different from your morphology. When we say architectural pattern, where are your cells supposed to be? Are your cells supposed to be arranged in a circular pattern? Are they supposed to be grouped together? Are they supposed to be far from each other, like in the case of your osteocytes in your bone tissue? Diba? Dikit-dikit ba dapat sila? Okay, hiwa-hiwalay ba? Naka-star pattern ba yan? May pattern ba sila? Alright, so you have to, that is what we call your architectural pattern. Okay, so your fibers, for example, are they supposed to be very close to your cells? Okay, are they supposed to be in a parallel arrangement, perpendicular arrangement? Are they supposed to be disarranged? So on and so forth. That is architectural pattern, which is entirely different from your morphology. Okay, nakuha? So again, there are two things that you have to preserve. It's the morphology as well as the architectural pattern of your tissue constituents. Okay? So in the process of preservation, you have to prevent, okay? You have to prevent or arrest degeneration or the degenerative processes. That starts upon blood supply deprivation, just like what I have mentioned a while back. Okay, so I repeat pag sinabi nating preservation, this involves preventing or arresting the degenerative processes which start upon blood supply deprivation. Okay, so yan. So that is the first purpose of your fixation. The second purpose of your fixation is the protection of your tissue samples. So when we say protection of your tissue samples, there is a need for you to adequately harden your sample. Nakuha? Ayan. Alright, so when we say adequately harden your tissue sample, okay, so this will make your tissue sample less vulnerable to the different damaging effects of the reagents that you are supposed to use in the subsequent steps in tissue processing. Nakuha? Remember, ano ba ang fixation? Ang fixation ay unang step pa lang yan. Okay, so there are still several steps that you have to perform in order for you to completely process your tissue sample. And in these different subsequent steps in tissue processing, there is a need for you to make use of reagents that have very high concentrations or very low concentrations. Okay, so some of these reagents might be detrimental to your tissue sample. So that's why you have to protect it. Okay, ayan. So apart from the chemical reagents that we use, there are also physical aspects of the tissue processing. Like for example, when you perform infiltration or embedding, this is carried out at very high temperatures. Okay, so that physical agent, yung very high temperature natin, it can be a physical agent that can possibly damage your tissue sample. Okay, another, when you perform sectioning, all right, so di ba pag sectioning, you are going to make very thin sections of your tissue sample. So again, this can be a physical, okay? This can be a physical means of possibly damaging your tissue sample. So in order to protect your tissue sample from the different chemical reagents and physical means of processing your tissue sample, you have to adequately harden it. So take note ha, I made use of the term adequately because we don't want naman our tissue sample to be over-hardened. Okay? Hindi naman pwedeng masyadong matigas ang tissue sample mo. Naku ha, ayan, adequate lang, yung tamang tigas lang dapat ang tissue sample natin. Okay? May tumatawa, may humingiti. Hindi pwedeng masyadong matigas. Dapat tamang tigas lang po ang kailangan. Naku ha, otherwise, hindi din maganda pag over-hardened ang ating tissue sample. Okay? So may I just go back to your preservation because I think I forgot to mention this. All right. So what again is this process that involves the digestion of your cells? I know you have encountered this term probably in your histology. Okay, what do you call this process that involves digestion of your cells? So, ang nagdadigest ay yung cell mismo. Dinadigest niya yung sarili niya. What do you call that process? Starts with letter A. We have, sige, bilis mga besh. Alright, yes naman. Miss Maderas. Okay, we have your otolysis. Okay, yes, Crossland. Thank you. So we have your otolysis. Okay. Ma'am, is otolysis different from apoptosis? Yes. Okay, yes, they're different. Naku ha, magkaiba po ang otolysis sa apoptosis. Pero 'di ba, we were taught kasi, actually ako din, nung una akala ko parehas lang ang otolysis at saka apoptosis. Because before, 'di ba, and in some reference really, or in some references, they use the terms otolysis and apoptosis synonymously or interchangeably. But actually, there's a difference between apoptosis and otolysis. So when we say apoptosis, this is normal. Okay, this is normally happening inside your body. It's part of the normal physiology in which cells are replaced by new ones. And apart from that, pag sinabi mo kung kasing apoptosis, it's a step-by-step gradual process inside your cell that takes place in a cell. Okay? Yan. So hindi siya yung katulad ng otolysis. Pag sinabi mo kasing otolysis, this involves tissue digestion by your intracellular enzymes which are released by your lysosomes, okay? By your lysosomes. Ayan, by your lysosomes. Alright? So, yung pag-release ng lysosomes ng ating mga enzymes, kalat-kalat. Okay, kalat-kalat. Bakit kalat-kalat? Kasi one of the first organelles which will be affected during autolysis is your lysosome. So in particular, your lysosome will undergo membrane rupture, kaya ganun. Okay? Minsanan niyang i-release yung kanyang lysosomal enzymes. And this will now cause, okay, this will now cause the autodigestion of your cells. Unlike pag apoptosis kasi, hindi siya minsanan na isang sabog lang ng enzymes. Pag apoptosis, gradual ang release, pakonti-konti. Okay? So, very systematic yung pag-digest ng cell. That's apoptosis. That's normal. But in autolysis, ano ulit? Okay? Minsanang release. So, that's why, okay, medyo mas, it's not organized as compared to your apoptosis. So, magkaiba yun ha. So, the correct term, therefore, that we should use in this case is otolysis and not apoptosis. I will not accept apoptosis as an answer. Naku ha? Kasi otolysis ang tamang term dyan. Alright? So, yan. So, apart from preservation and protection, your fixation also improves staining and optical differentiation of your tissue sample. Okay, so how will your fixation improve staining? This is because some of the fixatives that we use, so these are the chemical agents, okay, some of the fixatives that we use during fixation, they can serve as mordants. Nakuha, they can serve as mordants, thus improving the staining characteristic of your tissue sample. Okay, so apart from that, take note that it is important for you. Okay, it is important for you to consider the compatibility or incompatibility of your fixatives and your stain later on. So of course, hindi ko muna sasabihin, hindi ko muna i-elaborate ito because this is part of your QFR. Okay, so isa lang ang takeaway natin from this particular concept for now. So it is necessary for you to consider kung ano ba yung stain na gagamitin mo later on. Okay? Because it will significantly affect the quality of tissue sections that you are able to produce. Nakuha? So, syempre, kunwari, nagkamali ka ng fixative, andun ka na sa staining process. Do you think it's easy for you to remedy? Okay, if for example, mali pala yung fixative na ginamit, no. It's not that easy. Okay, yes. And in some instances, it might even require you a new sample. So that's why, okay, bago kunin yung tissue sample, okay, bago siya tanggalin doon sa ating dead body, for example, or during surgery, dapat alam mo na kung anong fixative ang gagamitin. Okay? That is compatible to your stain. Nakuha? So, yan. So, staining, that's also your optical differentiation. Parang halos parehas lang sila ng concept. Okay? Yan. Alright, so the reagents or the chemical substances that are utilized for fixation are referred to as your fixatives. So take note ha, the word fixative is only used for your chemical means of fixation. Hindi siya pwedeng gamitin for our physical means. Okay, so nagkakaintindihan na tayo dyan. Pag sinabi mong fixative, automatic, I'm referring to a chemical substance and not to a physical means of fixation. Like for example, heat. Can we call it a fixative? No, because that's a physical means of fixation. Okay, the use of your microwave, for example, no. We cannot use it as, or we cannot refer to it as a fixative. Because again, it's a physical means of fixation. Okay? How about freezing your tissue sample? Again, hindi pwede. Okay? Kasi it's a physical means of fixation. Alright? So, in the case of our fixatives, yes, how do they work? How do they work? So, primarily, their goal is to stabilize the different structures that are found in your tissue sample. Okay? So, paano nangyayari ang stabilization ng structures natin? Okay? So, dapat there should be cross links. Okay, hindi crossland there should be cross links that are formed between your proteins. Oh, bakit pumasok ang proteins dito? Okay, sige. Can you give me one important function of your proteins in relation to this discussion? Ano kaya yung isa sa mga importanteng functions ng ating proteins na related dito sa histopath? Saglit lang ha. Isara ko lang yung bintana kasi maingay talaga yung kapitbahay namin. Ayan. So ano kaya? Magbigay kayo. Isang function ng proteins na related dito sa discussion natin. Denaturation. Okay. Ano pa? Structure, framework of tissue. Okay. Okay. Tama, structure. Di ba you discuss this as part of your clinical chemistry and even your biochemistry? So one of the most important functions of your proteins is that they serve for structural purposes. Tama? Tama ba ako? Structural purposes ang proteins na yan. So, ibig sabihin, okay? Ibig sabihin ba, ma'am, ang ating mga cells ay made up of proteins? Yes. Okay? So, usually, in most instances, the backbone, or the framework of your cells, as well as the different structures which may be found in your extracellular matrix, they are protein in nature. Okay, so that means if you want to stabilize your tissue sample, if you want to preserve it, what should you target? It's the proteins. Nakuha? Ayan. So, ang ita-target mo dapat ay yung proteins. Kasi sila nga ang framework ng ating cells at saka ng ating tissue constituents. Okay? So, how are you going to stabilize them? By forming cross-links between your protein structures. Okay? So, in the formation of your cross-links, there are two mechanisms involved. So we have your additive, okay, and then the non-additive. Additive mechanism or non-additive mechanism. So these two terms may also be utilized in order to categorize your fixatives. Okay, so we have your additive fixatives, we have your non-additive fixatives. Nakuha? So from the term itself, pag sinabi nating additive, okay, so these fixatives will form cross-links by adding themselves to the link itself. So that means they become part of the link itself. Okay, so what are examples of fixatives which are considered to be additive in nature? So we have your aldehyde. Okay, we have your aldehydes, your mercury-based fixatives, as well as your osmium tetroxide. Okay, nakuha? So I repeat, we have your aldehydes, your mercury-based fixatives, and your osmium tetroxide-based fixatives. So those are good examples of additive fixatives. And then, we move on naman to the other one. We have your non-additive fixatives. So these fixatives naman, from the term itself, non-additive, they do not become part of the cross-link. However, in order for them to facilitate the formation of the cross-links, they have to remove water from the established crosslinks. Nako ha? Naku ha. So I repeat, it involves the removal of water in order to allow, nasa module ninyo, in order to allow new crosslinks to form. Okay? Yeah. So I think very example ng okay. So out of all the fixatives that we have, it's only your alcohol which is capable of non-additive mechanism. Okay, so that is a good example of a non-additive fixative. Naku ha? Ayan. Alright, so another way by which your fixatives can stabilize the structures is to make your tissue samples, okay, is to make, actually this is related to the first one, formation of your crosslinks, okay? Paki-erase na lang na another way. So, in short, saglit lang ha. So, in short, the ultimate goal, therefore, of forming your crosslinks is to make your tissue sample insoluble to the action of your lysosomes. Naku ha? So, I repeat ha. If crosslinks are formed, your tissue sample will now become insoluble to the action of your lysosomes. Okay? So, yan. Alright? So, again, the next one. Okay? Another important mechanism of your fixative is to inactivate naman your lysosomes. Okay? So, una, di ba, stabilization of your structures. Again, which will make your tissue insoluble to your lysosomes. So another mechanism or another mode by which your fixatives carry out the process of preservation is to inactivate your lysosomes. Okay? Yan. So it's either your fixative will make the structure stable or insoluble, or they may also inactivate your lysosomes, or both. Kasi may mga fixatives po tayo that are capable of performing both the stabilization of your structures and the inactivation of your lysosomes. Okay? So wow, isang ano pa lang yan. Okay. Anyway, okay lang yan. Anong pangalan yan? Slide pa lang yan. Okay, next naman tayo. Okay lang kayo ha? Ayan. Medyo dapat kasi naintindihan natin ang fixation. Okay, so next, we go to the different factors that retard the process of fixation and enhance the process of fixation. I'm not sure if this is in your module, so kindly check na lang. So if it's not, then of course you have to copy. Okay, so there are certain factors which will retard. Pag sinabi nating retard, it will slow down the process of fixation. Okay, and there are also some factors that we can consider in order to enhance the process of fixation. So when we say enhance, okay, so it will make the process more effective or efficient, or it can make the process shorter. Naku ha, ayan. Alright, so first, we have the factors that retard fixation. Number one, increase in size and thickness. Okay? So again, hindi pwede ang sagot na size and thickness lang. You have to qualify. What size and thickness are you referring to? So that's why do not forget the term increase in size and thickness. Okay, so I think this is somewhat self-explanatory. Of course, if a tissue sample is bigger or thicker, it will be harder for it to be fixed completely. Naku ha, mas mahirap siyang i-fix. Mas matagal siyang i-fix. Kasi nga mas malaki siya eh. Alright? So another, the presence of the following. So we have mucus. fats, and even blood. Alright. So say for example, okay, so say for example, kasama ka doon sa removal of a particular tissue from a patient, from a given patient. And of course, nakita mo, oh, ang dami niyang mucus. So what can you do in order to, yes, in order to prevent the mucus from interfering with the fixation process? So you can wash, okay, you can wash your sample in saline. Nakuha? Yes. So actually, this is also applicable if, for example, the sample is, or if the specimen is bloody. So for specimens that contain mucus and blood, you can wash the tissue sample in saline, and then after which you have to put it in your fixative. Okay, yes. So washing it in saline will, of course, remove these two agents, and then they will no longer be able to interfere with the fixation process. Okay? So what if naman your sample contains fats? So in order for us to more efficiently fix samples that are considered to be fatty, so we can cut them thinner, okay? We should cut them thinner and fix them longer. Nakuha? Ayan. So, class ha, pag sinabi mong retard, it does not necessarily mean na longer time. Pwedeng, ang sinasabi niya dyan is the quality of fixation. Nakuha? Ayan. Parang yung sa enhance din, sabi ko kanina, it does not necessarily mean na pag-enhance, it will make the fixation process shorter. Pwede din na it will make the fixation process more efficient. Okay, so yan. So in the case of your fats or fatty tissues, again, you have to cut them thinner and then you have to fix them longer for adequate fixation. So another factor that will retard fixation is a decrease in your temperature. So again, I will not accept answer na temperature lang. You have to qualify. What temperature are you talking about? So again, this is decrease in temperature. Okay? So of course, a decrease in temperature will affect the movement of your molecule. So we learned this in our general chemistry. So take note, okay? Take note na ang movement ng molecules at lower temperature is slower as compared to the movement of your molecules in higher temperature. So if your molecules are slower, okay, in cold temperatures, so this will, yes, this will interfere with the penetration of your reagent in your tissue sample. Okay, syempre pag mas mabagal siya, mas mabagal din ang, or mas mabagal ang movement rather ng molecules, mas mabagal din ang penetration nila sa ating tissues. Okay, so yan. And then, let us consider factors naman that enhance the fixation process. So number one, we have decrease in size and thickness. Okay, decrease in size and thickness. So again, if it is smaller or thinner, it will be faster for you to fix it. And of course, the fixation process is also more efficient. Okay? The presence of agitation as well as heat or increase in temperature. So both of these, okay, both of these factors will serve to increase the movement of your molecules. So again, if the movement of your molecules is increased, so this will make the penetration of your tissues or of your fixative rather into your tissue sample more efficient. Okay, or faster in that case. So, 'di ba, pag mas ang mabagal, mas mabilis ang movement ng molecule, syempre, mas mabilis din yung penetration niya. Okay? However, you have to put an asterisk on heat. Okay, lagyan nyo ng heat dyan. Lagyan nyo ng asterisk yung heat. Why? Because, take note ha, moderate heat. Okay, so when we say moderate heat, this ranges from a temperature of 37 to 56 degrees Celsius. Okay, so I repeat, moderate heat. So in particular, we are talking about 37 to 56 degrees Celsius. You have to take note that at this temperature, autolysis also is faster. Okay, so moderate heat therefore also hastens the autolytic changes and enzyme destruction that takes place in your tissue sample. Okay, so how will this affect now our decisions in the fixation process? So pag maliit at saka manipis yung tissue sample natin, doon lang natin pwedeng i-consider ang moderate heat. Naku ha, I repeat ha, pag manipis at maliit ang ating tissue sample, doon lang natin pwedeng i-consider ang heat. However, if you are tasked to fix an entire organ, so say for example, ganyan, isang buong heart ang i-fix mo. Okay. So do you think increasing the temperature will fix your heart sample faster entirely talaga as compared to it undergoing autolysis and destruction? Okay, hindi pwede. Hindi pwedeng gumamit ng heat. Naku ha, pag ang pinag-uusapan natin ay entire organ. Why? Because, remember, ano ang penetration, ano ang best penetration rate ng mga fixatives natin? Okay, ayan. Kasama yan doon sa unang discussion ko. What is considered to be the fastest fixative? Ano ang kino-consider natin na fastest fixative? Anybody? Okay, formaldehyde. Yes, thank you. So ano ang penetration rate ng formaldehyde? Ano ang penetration, Toroy? Ay hindi naman, yeah, tama, actually tama din naman si Crossland. We have acetone, yeah, it's actually the fastest. Okay, pero sa mga apart from your acetone, okay, kasi walang binigay na penetration rate for our acetone. So ano yung best na penetration rate natin? Okay, yes, so we have your 1 millimeter per hour. Take note of that penetration rate, 1 millimeter per hour. So just imagine kung buong heart ang kailangan mong i-fix. Okay? After ilang oras, naku, wala pa siya doon sa gitna ng heart. Tama ba? Tama ba ako? Okay? So ibig sabihin, pwedeng yung outer layers ng heart mo, it will be adequately fixed. However, how about the inner portions of your heart? Diba? So, pwedeng yung inner portions ng heart sample mo, it has undergone significant destruction already, significant disintegration of your cells and your tissue components bago pa ma-reach ni fixative yung kaloob-looban ng tissue sample mo or ng organ mo for that matter. Naku ha? So that's why we should not make use of heat when we are fixing an entire organ for that matter. Kasi oo naman. Mafi-fix niya yung outer portions. But how about the inner portions? Okay? Yes, yan. So that's why I mentioned that heat will actually make your autolysis and enzyme destruction faster. Okay? Yes. So, dapat i-consider din natin yon later on. Alright? So, those are the different factors that hasten and retard the process of fixation. So now, let us consider yung mga factors that are needed in order for us to perform the process of fixation given a specific sample. Okay, yes. So we have to consider kasi pag pina-perform natin ang mga procedures natin in the lab, what type of microscopy are you going to use? Okay, is it your light microscopy or is it your electron microscopy? Okay, so apart from that, what specific structure are we supposed to study, or does the pathologist rather supposed to study? Okay, so yan. So those are the different considerations. Okay, yes, that we will be discussing for this portion. Okay, so number one factor that we have to consider, we have your pH. So take note, class, that pH, this is your power of hydrogen. It is otherwise referred to as your hydrogen ion concentration. So the optimal the optimum pH of your fixatives, it has to be 6 to 8. Okay, it has to be 6 to 8. All right, so like for example, in the case of your formalin, di ba, it has to be maintained at a pH of 7. Okay, so between acidic and your basic pH, we prefer a basic pH. Naku ha, so I repeat so if you are to choose between acidic pH or basic pH, mas kino-consider natin ang basic pH. Why? Because remember, 'di ba? Ano ang unang nawawala pag tinanggal mo ang tissue sample sa loob ng katawan? You deprive it of blood supply. Tama? Blood supply deprivation. So one of the nutrients, okay, which are not supplied to you, or which are not tuloy, one of the nutrients which is not supplied to your tissues once it is removed from the body is your oxygen. So this will now cause, okay, ano ang term na ginagamit natin? Pag mababa ang oxygenation ng ating mga tissue samples or ng ating mga cells. Okay, yes, very good. So we have the term hypoxia. Okay, so remember if a particular tissue or a cell rather undergoes hypoxia. Okay, so ano ang what metabolic pathways are performed by that particular cell if oxygen is not available? If oxygen is not available, what metabolic pathways can the cell perform? Ala, nawala na yung mga video nyo. Okay, anaerobic. Oy, dalawa lang ang nagre-recite Grabe, hindi ko ibibigay itong record na to pag walang nagre-recite. Okay, so ang fine-favor, 'di ba? Therefore, na pathway ay anaerobic. And we all know that anaerobic metabolism leads to the formation of your acids. Tama? Okay. So hypoxia therefore will ultimately lead to acidity. So the problem here is, especially if you are using formaldehyde or formalin as your fixative, acidity favors the formation of your formalin heme pigments. Naku so I repeat acidity leads to the formation of your formalin heme. Hindi heme na HIM Sige, lagay ko. We have formalin heme pigments. Okay, yan. So your formalin heme pigments, these are black polarizable deposits which may be seen in your tissue sections. Okay, so of course, since they become incorporated in your tissue sections, they can interfere with the interpretation later on, which will be made by the pathologist. So pwede din that they will obscure significant structures which are supposed to be examined or viewed by your pathologist. Okay, so yan. So that's why, as much as possible, we don't want an acidic pH. Okay? However there are exceptions to this rule. Kasi may mga, may mga fixatives tayo which are really supposed to be acidic in nature. Okay? So another factor that we have to consider is the temperature. Okay? So temperature naman tayo. So it depends upon the type of fixation that you are performing. So for routine manual, okay, for routine manual tissue processing, the temperature should be 20 to 22 degrees Celsius. So this temperature has been described by Gregorius as room temperature. Okay, so I repeat ang room temperature pag fixation ang pinag-uusapan natin ay 20 to 22 degrees Celsius. So please take note of this. Kasi iba din ang room temperature pag decalcification naman ang pinag-uusapan natin. Okay? So yan. So routine manual yan. And then for our routine automatic processing. Okay? Yes. Bakit magkaiba sila? Kasi remember na ang ating automatic processing, so yun na ang nagiging trend in our laboratories, particularly in histopath. Okay, so it has become a trend already. And apart from that, parehas silang routine. Naku parehas po silang routine routine manual, routine automatic processing. So that means, hindi porket automatic processing siya, hindi na siya routine. It's still considered to be routine because we are performing the same steps naman. But this time, with the use of your automatic processor. So automatic processors, they are maintained at a temperature of 40 degrees Celsius. Okay, 40 degrees Celsius. For electron microscopy as well as for your histochemistry, so it has to be, the fixation process has to be performed at 0 to 4 degrees Celsius with the exception of your mast cells. So if for example, may tissue ka, may tissue specimen ka, and the pathologist would like to see mast cells, okay, using electron microscopy as the method of examination. So, ibig sabihin, hindi ka pwedeng mag-perform ng fixation at 0 to 4 degrees Celsius. Why? Because your mast cells are preserved or are best fixed at room temperature. Naku so depende din yan. Okay? Yes, depende din. For the other electron microscopic study of the other structures, so you perform it at 0 to 4, except nga pag mast cells ang pinag-uusapan natin, ano ang temperature dapat? We have your room temperature. Okay? So another, you can also heat your formalin or your formaldehyde at 60 degrees Celsius. So formalin at 60 degrees Celsius, this is in general used for the rapid fixation of very urgent biopsies. Okay, so I repeat, rapid fixation of very urgent biopsies. You can increase the temperature of your formaldehyde solution to 60 degrees Celsius. Okay, so formalin naman heated at 100 degrees Celsius is utilized for the fixation of tissues that contain your tuberculosis causative agent. Okay, so yan. So tissues with the tuberculosis pathogen, so we can make use of formalin at 100 degrees Celsius. So take note meron po tayong extrapulmonary tuberculosis. So that means, okay, your Mycobacterium tuberculosis does not only affect your respiratory system or your lungs for that matter, it may also affect other organs. And as such, we can make use of the term extrapulmonary tuberculosis. Okay? So another is in the case of your nucleic acids. Okay, sige. Dahil napagod akong magsalita, ano ang structure ng ating mga nucleic acids? Ano? Ano na? Long chain-like structure, branched. Hala kayo! Ibabalik ko kayo kay kay biochemistry. Susumong ko nga kayo sa mga teachers nyo. Okay, yes, but what is the structure? Anong structure ng ating mga nucleic acids class?
Double helix.
Double helix. Okay, that is in the case of what?
DNA po.
DNA. How about your RNA, Lawrence?
Single helix.
Okay, single helix. So in short, parehas silang helical in shape. So another tuloy. Probably a simpler way to describe our nucleic acids is that they are coiled. 'Di ba? Naka-coil sila, tama? Paikot sila. Okay? So for your RNA, it's single. For your DNA, it's double helix. Tama? Okay. So since they are in a coiled structure, they are helical in structure, formalin at the routine temperature that we use cannot properly fix your DNA and your RNA. Okay? Your nucleic acids class, they do not react with your formalin at room temperature. Kasi nga helical sila, coiled sila, so maarte po sila. Okay, so in order for us to allow your formalin, for example, to react with your nucleic acids, you have to uncoil your nucleic acid first. Okay, and what will uncoil your nucleic acid? It's an increase in temperature. Nakuha, increase in temperature leads to the uncoiling of both your RNA and your DNA. So since your RNA is single, mag-isa lang niya, okay, will it be easier to uncoil your RNA? Yes. That's why we make use of a lower temperature, and that is your 45 degrees Celsius. Okay, your DNA, since it is double helical or double helix, it needs a higher temperature. In particular, we have 65 degrees Celsius. Okay, nakuha. Ayan ha, do not forget. Okay, so next na po tayo. Naku, bilisan natin kasi nagugutom na ako. Okay, another set of factors that we have to consider would be the size and thickness of your tissue. Class, okay lang ba na ganito ako magsalita? O nababagalan ba kayo? Gusto nyong bilisan ko ng konti or okay lang ba? Okay lang ba? Mag-react kayo.
Okay lang po, ma'am.
Okay lang po, ma'am.
Okay.
Okay lang po, ma'am.
Okay, sige, okay lang. So, size and thickness of your tissue sample. So, again, it depends on whether you are going to perform light microscopy or electron microscopy. Okay? So, for light microscopy, yes, you may use large, hindi naman larger, but yeah, basically yun naman ang ibig sabihin niya. So you can make use of a larger tissue sample, okay, or tissue cut. So specifically the area is 2 square centimeters by 4 millimeters thick. Okay, so pag light microscopy yan. For electron microscopy naman, we make use of a smaller sampling size. So we have 1 to 2 square millimeters. Okay, 1 to 2 square millimeters for our electron microscopy. For the thickness, particularly if we're talking about light microscopy, so there is an exception to this. And the exception is in the case of your lung edema. Okay, in the case of your lung edema. Actually, this is the reason why ayoko sanang nag-Gmeet kasi mas prefer ko talaga ang Zoom. Hindi ko kasi alam gamitin yung pen tab ko. That's why I cannot write. Unlike pag Zoom siya, nakakalagay ako dyan ng mga ano. But anyway, here we are. We cannot do anything about it. Do not cry over spilled milk, Mom Peewee. Okay, so anyway, lung edema, the sample for this one has a tendency to collapse. Okay, so that's why if you are handling lung edema for light microscopy, it should be thicker. Okay, so the thickness is 1 to 2 centimeters po. Ako ha, so I repeat ha, pag lung edema, 1 to 2 centimeters thick dapat po siya. Okay, so next we have your osmolality. Alright, so osmolality, you can make use of the word osmolality or tonicity. Okay, tonicity or osmolality po. Which one pala is a broader term? Mas broad ang osmolality ha, or osmolarity. Okay, in histopath kasi hindi naman medyo, but they make use of the terms osmolality or osmolarity interchangeably. Okay, yes. It's not very strict when it comes to the use of the terms osmolality or osmolarity. Unlike when you had probably your analytical chemistry. Okay, yes. May difference, 'di ba? Ang molality at saka molarity. Okay, but yun nga, sa histopath, hindi natin siya talagang fina-follow yung strict definition ng mga words na to. Okay? Yan. So yan. Anyway, ano ang mas broad na term? Osmolality or tonicity? Hello! Ano ang broader na term? Osmolality or tonicity? Mga besh. Hoy, bilisan nyo. Sumasakit ang batok ko. Baka ma-high blood ako dito. Okay, yes. So we have your osmolality. Okay, so take note ha, na pag sinabi nating osmolality, we are considering the concentration of your solutes in general. Okay, pag sinabi mong tonicity class, the solute that we are considering is your sodium chloride. Okay, nakuha. Pag osmolality, solutes in general. Pag tonicity ang pinag-uusapan natin, we are considering the concentration of your sodium chloride, okay, in a particular solution. Alright, so osmolality, this is actually not a very strict rule when it comes to the fixation process, okay? Not a very strict rule when it comes to your fixation process, okay? One thing that you have to consider for your osmolality would be the reaction of your cells, okay, or the effect of such osmolality to the morphology of your cells, okay? So for hypertonic solutions, diba? For hypertonic solutions, so this will cause your cells to shrink. Okay, hypertonic, shrinking of your cells, or your cells will become smaller. Okay, in contrast, if we consider a hypotonic solution as well as an isotonic solution, your cells will swell or they will become larger. Okay, so this is in the case of histopath ha. Baka mamaya you will argue na naman na, but ma'am, we learned in biochem that isotonic is not supposed to change. Okay, the morphology of cells. Well, that's true. That's true. Okay. However, in the reference that we use for histopath, it says there na ang isotonic solution will cause your cells to swell. Okay, yes, it will cause your cells to swell. So that's why if it is asked in histopath, swelling or the cells will become larger ang tamang sagot. Nakuha because it's in our reference. Okay? So yan. So that's why between hypertonic and your hypotonic solution, there should be a balance. Okay? There should be a balance. So ang gusto talaga natin, okay, ang gusto talaga natin, it depends pala on the type of study that you are going to perform. Okay? So if you are going to perform light microscopy as your method of examination, the preferred osmolality is slightly hypertonic. Okay? But if you are going to make use of electron microscopy as, okay, electron microscopy as the mode or method of examination, the preferred, okay, the preferred tonicity is isotonic. Okay, so I repeat, pag light microscopy, it's slightly hypertonic. Pag electron microscopy naman, it's your isotonic. Okay? Bakit kaya magkaiba sila? So this has something to do with the effect of the movement of your molecules. Okay? So remember ha, 'di ba ang cells kasi natin, they are inherently isotonic. Tama? Okay? So iso yan. All right. So if there is a greater difference between the intracellular environment and your solution, okay? So this will create a faster movement of your molecules. If you can still remember from your biochemistry, we call this your gradient. Diba? Naalala nyo pa ba ang gradients natin? Pag sinabi mong gradients, okay, this pertains to the difference of your solution to that of your cell. Okay? So, pag light microscopy ang pinag-uusapan natin, kahit na mabilis yung movement ng ating molecules, okay lang. Kahit na mabunggo-bunggo ni molecules yung mga details ng cells natin, okay lang. Okay? Bakit? Kasi hindi naman siya mamamagnify ng sobra-sobra. So that means, yung minute details, yung mga very minute details ng ating mga cells, they are no longer seen in your light microscopy. Tama ba? As compared, pag ang method na gagamitin mo ay electron microscopy. So sa electron microscopy, since your image is magnified several times more as compared to your light microscopy, there is a need for you to preserve even the minute details. Okay? So, ibig sabihin, yung movement ng molecules natin, yung pagbunggo-bunggo ng molecules natin doon sa mga structures ng cells, it will affect, okay, the image that is created if we are using electron microscopy. Nakuha ba? Nakuha? Kung bakit sa light microscopy ay slightly hypertonic and then sa ating electron microscopy ay isotonic dapat. Nakuha ba? Ay, bakit nyo ako iniwan? Ang dami nagli-leave. Bilis. Sabihin nyo sa akin kung nakuha nyo. Ito ang ayoko sa live hindi Okay, yes, nakuha. Yes, thank you, Nicole. Ayan. May lag talaga ang mga internet connections natin. Okay, so anyway, so I do hope you were able to get that. Kung hindi nyo nakuha, ulitin nyo ulit kasi recorded naman to. Alright, so in the case of your electron microscopy, yes. So, since one of the solutions or fixatives that is used for electron microscopy is your osmium tetroxide, okay? So we maintain or we achieve the right osmolality of your osmium tetroxide for electron microscopy by adding sucrose. Okay, yan. So how do we achieve? Yes, in the case of your osmium tetroxide for electron microscopy, we add sucrose. Okay? Yan. So another, by the way, ang isotonic has an osmolarity or osmolality of 340 milliosmoles. Okay, pag electron microscopy, ideally, it should be more or less isotonic. So, ibig sabihin, hindi greater or less than isotonic yan na. Ibig sabihin po ng phrase na more or less ay malapit siya sa isotonic. Okay, that is for your electron microscopy. Alright, so next factor that we have to consider is your formaldehyde. So, in the case of your formaldehyde, the routinely used concentration is 10%. For your glutaraldehyde, on the other hand, which is utilized for electron microscopy, so for routine electron microscopy, we make use of your 3%. Okay? And then for your immunoelectron microscopy, we make use of 0.25% glutaraldehyde. So what does this tell us? So it depends upon the fixative that you are going to use. And apart from that, it depends upon the specific study or type of examination that you're going to perform later on. Okay, so this will, yes, determine the concentration of your fixative that is to be utilized for a certain given or for a certain sample in the laboratory. Okay, so another factor that we have, another set of factors, medyo marami po ito, we have your volume. Okay, and then the last one, we have your duration. Yes, napaka-perfect ko talaga. Ilan ang natapos kong slides? Oh my God, 5. Very good. Okay, so we have factors to be considered. The first one is your volume. Alam ko, siguro yung iba kumakain na sa inyo, no? Bakit nag-off na kayo ng camera? Or baka mabagal lang yung net ko. But anyway, volume. Okay. So for routine fixation, hindi ako nagkamali dyan ha, 10 to 25 times the volume of your specimen. I usually read the second edition of Gregorius. Why? Because the second edition is better than the third edition. I don't know why. Pero, out of all the editions of your Gregorius, okay, ang pinakamaganda talaga is the first edition. Yeah, I don't know why. Yeah, but this is also according to my instructor before. Ang pinakamaganda talaga is the first edition. Okay? Diba, editions are supposed to become better, but yeah, I don't know. Not in the case of your Gregorius. Pero syempre, ang good thing kasi sa mga newer editions ng Gregorius is that mas maganda yung kanyang discussion ng histochemistry. Okay? Yun yung na-appreciate ko dito sa third edition ng Gregorius. But if you are going to compare, better pa din yung mga older editions niya. Okay? So that's why if you will be able to have a copy of the older editions, yeah, maganda siya. Okay? Ayan. So anyway, volume. In the second edition of your Gregorius, it was mentioned there 10 to 25 times. Okay, so that is the acceptable volume of your fixative. So ano ang unit dito? Times the volume of the specimen. Okay, so that's why before, nung face-to-face pa, I do not accept answers na 10 to 25 times lang. Times what are you talking about? Okay, so dapat kompleto 10 to 25 times the volume of your specimen. So without the volume of your specimen, I do not consider that answer because it's incomplete. Okay? However, if you are going to look at the volume, the optimum volume of your fixative, it's 20 times. Okay? It's 20 times. Nakuha? So, for example, in the exam, given lahat, 10 times, 15 times, 20 times, 25 times, the best answer is your 20 times because that's the optimum volume. Okay, that's the optimum volume. Now, for prolonged fixation, such as in the case of your museum preparations, so the required volume of your fixative is 50 to 100 times the volume of the specimen. Okay? Ayan po. So, in the case naman of your osmium tetroxide, so remember, your osmium tetroxide is utilized for electron microscopy. And apart from that, your osmium tetroxide is an expensive fixative. So that's why we use it to a minimum volume. Okay, so the required volume for your osmium tetroxide is 5 to 10 times the volume of the specimen. And then lastly, so last na po muna ito, and then I will probably record na lang the next part of the introduction. So the duration of fixation, it depends. Okay, it depends upon the type of examination that you will perform later on, and apart from that, it depends upon the fixative that you are using. So say for example, in the case of your formaldehyde, for routine tissue processing, the duration should be 24 hours. Okay? For our electron microscopy naman, so the duration is 3 hours. Okay? So I know, di ba hanggang 7 lang tayo pag TTH? Tama ba? Thursday ngayon, di ba?
Yes, ma'am.
Okay. O, di ba? Pag dismissal, ang bilis mag-reply. Okay. So anyway, so since we don't have time na, so I will just be cutting our discussion here and then record na lang the rest of the introduction. Baka bukas ko na ito mai-record. Bukas ng madaling araw. And then, anyway, pag naman, konti lang naman na to. Hanggang organ preparations lang naman. Ay, characteristics pala of a good fixative. Kasama pa rin po sa quiz, ma'am. Oo, lahat. Kasama yan. Of course, kasama yan. Lahat. Magbo-bargain na naman kayo. Wait lang. Pagpahingain nyo muna ako para bumait-bait ako. Okay? Wala mo nang magbo-bargain ngayon. Okay? Pagpahingain nyo ako muna para bumait ako. Alright. So, do you have any questions? From first video po, ma'am. Opo, Miss Gaspar. Yes, lahat yan. Buong fixation. Okay, so yan. Mamaya na lang tayo or bukas na lang tayo mag-usap. Basta, ang ating coverage ng quiz ng Saturday, basta, bukas na tayo mag-usap. Okay? Naku ha? Ayan. Yeah, rest muna tayong lahat para mahimasmasan ang ating mga utak at ang ating mga puso at nang bumait-bait po tayong lahat. Okay? So, thank you so much for attending. Happy dinner to Carmeline. Okay? So, thank you so much for attending our synchronous class. So, again, if you have further questions or any questions, so you can raise it in our group chat. So, thank you and have a great evening. Yes, eat your dinner. Yes,
Thank you,
bye.
ma'am.
Thank you, ma'am.
Bye.
Thank you, ma'am. Bye.
Bye. Bye.
Ma'am,
Yes.
Thank you po, ma'am.
ang wala brown out kasi.
Ay,
Anong brown out? Basta nga, wait lang kayo. Basta. Brown out. Anong brown out? Anong brown out?
Sa amin na.
Si Resurrection Burnout, ma'am.
Ewan ko sa inyo. Oh, sige. Resurrection, brown out. Sige. Basta. Kailan brown out ngayon?
Ma'am, ngayon po.
Ngayon, brown out ngayon? Okay, sige. Ala, kawawa ka naman.
Umuulan po, ma'am.
Oo. Kasi rainy season na. Sige, basta pag-usapan muna natin. Basta, you do your best to prepare. Yun na lang muna. Mag-aral muna kayo. Para at least, kung kunwari man na buong fixation ang quiz sa Sabado, di at least ready na kayo. Okay?
ma'am,
Yes.
ma'am, question ko lang po sa may
Yes po.
QFR.
Yes
Sa
po.
may decalcification po.
Yes.
Sabi po kasi dito, what are the precautionary measures and guidelines in decalcifying tissues?
Mm.
Ma'am, yung buong process po ba ilalagay, ma'am, or yung like guidelines lang po?
Guidelines lang, oo. Kasi 'di ba nung inexplain ko siya, sabi ko, you have to consider yung proper decalcification ng tissue sample. Okay? Hindi naman niya tinanong kung discuss the process, hindi naman ganun eh. So, what are you supposed to consider lang? Like for example, like for example, the size of your tissue, ganyan.
Ay,
Okay,
parang
mga ganon.
temperature po, ganun
Oo,
po.
yeah, correct, Besh. So,
Thank you, ma'am.
okay.
Sige
O,
po.
babay na ha. Kakain na kami.
Ay, ma'am.
Yes,
Thank you
ma'am.
po.
Bye.
Thank you po, ma'am.
Yeah, bye.